Good day, everyone. This is part 1 of our lecture on medicinal chemistry. This is the outline of our video. First, we will discuss what medicinal chemistry is. We will define what a drug is. We will review proteins, amino acids, and binding. We will discuss drug targets, drug design, and drug development. Okay, now let's start. The subject of medicinal chemistry explains the design and production of compounds that can be used for the prevention, treatment, or cure of human and animal diseases. Medicinal chemistry includes the study of existing drugs, specifically their biological properties and their structure-activity relationships. It concerns the discovery, the development, the identification, and the interpretation of the mode of action of biologically active compounds at the molecular level. Medicinal chemistry covers the following stages. First, in the first stage, new active substances or drugs are identified and prepared from natural sources, organic chemical reactions, or biotechnological processes. They are known as lead compounds. The second stage is optimization of the lead structure to improve potency and selectivity and to reduce its toxicity. And the third stage is the development stage, which involves the optimization of the synthetic route for bulk production of the active substance to render it clinically useful. Medicinal chemistry is almost always geared toward drug discovery and development, and these chemists work with a team of scientists from different disciplines, including biologists, toxicologists, pharmacologists, theoretical chemists, microbiologists, and biopharmacists. Together, this team uses sophisticated analytical techniques to synthesize and test new drug products and to develop the most cost-effective and eco-friendly means of production. Now let's define what a drug is. To be clear, we will mostly discuss medicinal drugs or pharmaceutical drugs throughout our discussion in medicinal chemistry. A simple definition of which is any compound that has a desired biological effect on the human body or some other living system that is intended for the diagnosis, cure, treatment, or prevention of a disease or discomfort. Okay, side note, when introducing such compounds in our bodies, there is a thin line between it being therapeutic and being toxic, especially in the amounts administered in the body. But more on that when we discuss toxicology. Now we are done with the first 2 points in the outline. Before we go to a new concept entirely, let's first review something you might have heard of before that would really help us understand more complex concepts in medicinal chemistry. We will revisit none other than proteins and their binding interactions because they play a crucial role in drug action. Okay, now in this slide, we're only reviewing the basic concepts of proteins. Proteins are, if you recall, made up of amino acids with side chains. Side chains can be charged either positive or negative, or side chains can also be uncharged and can be polar or non-polar. You might want to review the list of 20 amino acids found in proteins. Also, recall that the tertiary structure of a protein, which refers to its three-dimensional shape, is largely determined by the interactions between these side chains. Proteins fold in a specific way to maximize attractive forces and minimize repulsive forces. So in this illustration, if you notice, you will see most of the polar and ionic side chains on the surface of the protein that are exposed to water. On the other hand, non-polar side chains like methyl groups and aromatic rings are clumped together, hiding from the water as much as possible. Kaya ayaw nilang ma-expose sa aqueous environment. Consequently, This folding may give rise to pockets on the surface of proteins that are potential binding sites. Take note that these binding sites are very important because once they find a molecule that fits this binding site, as you can see here, a biological response may occur. And take note of the change in the shape of the protein here. It kind of follows the contour of the introduced molecule, similar to a hand gripping an object. You might even see it more clearly in this slide, where you can see a much visible adjustment of a protein around an introduced molecule. Note that our protein is not rigid in structure. Rather, it is flexible enough to adjust its shape. And this is in order to maximize the attractive forces of the molecule with the side chains of the amino acids at the binding site. And when attractive forces are maximized, the more stable it is. So this is the concept of our induced fit. Now, speaking of binding, there are two ways a molecule can bind to a protein in its binding site. It may either be reversible or irreversible. Reversible binding interactions involve your intermolecular forces of attraction, such as London dispersion forces. So these are attractive forces between nonpolar molecules. In the context of proteins where they are found in our bodies and are expected to be in an aqueous environment, London dispersion forces are also referred to as hydrophobic interactions. This simply emphasizes that your nonpolar molecules clump together because they want to minimize their exposure to water, as mentioned earlier. A specific type of London dispersion force, which might be new to you, is pi stacking. And this is specifically observed in aromatic rings. There is a certain stability achieved when their pi electron orbitals overlap. So this is also an important hydrophobic interaction that to look out for. Next, dipole-dipole forces. These occur between polar molecules. Hydrogen bonding, you already know this for sure. These are special dipole-dipole forces between a hydrogen and an electronegative atom such as oxygen and nitrogen. Ionic interactions, these are, of course, from the name itself, between positive and negative charges. And lastly, if we have reversible binding, then we also have irreversible binding, which is much stronger than all that we've just mentioned. So these are your covalent bonds. Now that is it for the review of our proteins. So according to our outline, we will go to the next bullet point, which is drug targets. So we will focus mainly on receptors and enzymes, but I will also show you a bit of nucleic acids as drug targets as well. First, let's define what a drug target is. A drug target is simply any molecule that a drug interacts with to produce a therapeutic effect. Here, we have a pie chart to break down the various drug targets, illustrating the diversity of molecular sites that drugs interact with. A large portion of drug targets are your receptors. Top 3, which are colored green, red, and yellow, are all under this category. You have here G protein-coupled receptors, ion channels, and nuclear receptors. We also have the other category represented in white. A large part of this is your enzymes and nucleic acids. Again, we're not going to discuss all of the possible drug targets, but we will limit them to receptors, enzymes, and nucleic acids. So let's start with the first one, which are your receptors. Receptors are specialized proteins found on the surface of cells or within the cells that bind to chemical messengers. And when a chemical messenger fits in the binding site of a receptor, a quote-unquote message is sent out. So this message is some biological response. Then, afterward, the messenger leaves the binding site. You may think of the receptor as a faucet that a cell wants to turn on or off. So the binding of the messenger to the receptor makes the turning on and off possible. Okay, more details in this slide. Receptors are responsible for the communication between cells and regulating important cellular processes. Receptors have a binding site activated by chemical messengers such as hormones or neurotransmitters, and when inactivated, you get a biological response such as muscle contraction, transcription in the nucleus, and other sorts of responses. There are 4 common types of receptors. I'm listing them here so you can be familiar with these terms. First, we have ion channel receptors. Second, G protein coupled receptors. Next, kinase linked receptors. And lastly, nuclear receptors. The first 3 receptors are found in the cell membrane. Ion channel receptors act like gates of the cell membrane. Activating it will allow ions to pass through it. G protein receptors and kinase linked receptors are a bit more complicated because these involve multiple steps before you achieve a particular cell response. Lastly, the nuclear receptor is different out of all of them because it is not found in the cell membrane but rather in the nucleus or cytoplasm. And upon activating a nuclear receptor, the response directly influences gene expression. Now, if a drug can fit in the binding site of a receptor, then it can provide a way to modulate or alter normal physiological processes, allowing us to treat diseases by influencing cellular function. To visualize it at a molecular level, take a look at the illustration. A receptor with a binding site that has regions for H bonding, London dispersion, and ionic interaction ideally will interact with a molecule that has functional groups that are compatible with the binding site. Note that you can change some parts of the molecule. Hindi naman exactly ito lang ang fit na molecule sa receptor. So as long as it can interact correctly with the receptor then there can be a response. Okay, now for a drug to be effective, it should bind strongly enough to the receptor and thus trigger the desired effect. So the more binding interactions, the better. However, we don't want it to bind too strongly because it could block the binding site, interfering with the receptor's natural function. And usually that is harmful. So ideally, a drug should bind reversibly so it can leave the binding site unchanged. So in this slide, I will be introducing 2 new terms, agonist and antagonist. So under agonist, we have full, partial, or inverse agonist. But let's take a look at this video clip first so you can take note of the definitions.
Drugs on receptors can work as an agonist, a partial agonist, an antagonist, or an inverse agonist. Let's learn these concepts with an analogy of tap. Let's get started. Most of the receptors in our body are like taps that we see at public places. These taps stay open only as long as you keep the button pressed. When you release the button, the tap closes. Let's say this tap is for drinking water in a park. A man wants to drink water. He presses the button to the maximum so the water flows at maximum possible rate. Then comes a kid who also presses the button. But he is not strong enough to press the button to the maximum. So the water flows, but not at the maximum rate. See, the man is present and he is capable of opening the tap to the maximum. But as long as the kid occupies the tap, he cannot do so. Now let's say a caterpillar comes here and sits on the button. It cannot open the tap. But as long as it stays there, neither man nor the kid will be able to open the tap. In this analogy, the tap is a receptor. Most receptors in the body are like such taps. They are totally inactive under resting conditions. the man, kid, and caterpillar are ligands. The man is a full agonist. It activates the receptor to its maximum capacity. A partial agonist is like this kid. It activates the receptor, but not to its maximum capacity. Also, it prevents the activity of the full agonist. An antagonist is like a caterpillar. It binds with the receptor but does not produce any effect. However, it prevents the activity of agonists. Now, what then is an inverse agonist? Well, the concept of an inverse agonist is applicable for only those receptors which are constitutively active. Like this leaky tap, the water is flowing out even when no one is acting on this tap. It means the tap is active even in its resting condition. Here, that man can press the button to increase the water flow. But another possibility is a watchman of the park can come and lift the button to stop the flow. Here the watchman is an inverse agonist. Water is flowing at resting condition and in the presence of an agonist. But the watchman is stopping the flow. The caterpillar, as mentioned, is an antagonist. When it is there, neither man nor the watchman will be able to do anything. The tiny amount of water will keep leaking as long as it stays there on the button.
Okay, now that you've taken a look at the video, let's define some terms. An agonist is a molecule that binds to a receptor and activates it, triggering a biological response. For a drug to be an agonist, the drug must have correct binding groups, and the drug must have these binding groups correctly positioned. And the drug must be in the right size for the binding site. An antagonist, on the other hand, binds to a receptor but does not activate it. Instead, it blocks the receptor or prevents the agonist from binding and triggering a response. Thus, antagonists tend to bind more strongly than agonists. Now, take a look at this graph that shows the change in cellular response when a drug is administered. And let's assign the 4 of these. Which cellular response corresponds to each? At 0, which is the baseline, we have here the receptor only. It is currently functioning in normal conditions without external influence, so this is the baseline. Now, with an antagonist, it's the same thing. The change in response is still 0. The receptor still functions at its baseline, but the antagonist will hinder any change from now on because it is blocking the binding site. But then, in the presence of an agonist, the change shoots up to 100%, completely inducing a response. It is undeniably, without a doubt, na activate C receptor. If the change in response is less than 100%, then we attribute it to a partial agonist. It activates the receptor to a lesser extent, so less than 100% yung change, but it's still a positive value. And lastly, we have a response that is lower than the baseline. This is attributed to an inverse agonist. So this is the case when a receptor has some kind of function on its own, and that continuous function is its baseline. So once na may inverse agonist, then the receptor is completely turned off at negative ang change sa response. Now, that is it for receptors as drug targets. Let's move on to the second one, which is your enzymes. Enzymes are also proteins, but unlike receptors, enzymes can be found inside as well as outside of cells. They are usually free-floating in our body fluids. Enzymes act as catalysts, meaning they speed up reactions in the body. Okay, now, how does a catalyst help in chemical reactions? Okay, recall that in a chemical reaction, you begin with reactants, okay, and you convert them into products. So for the reaction to occur, the reactants need to overcome a certain activation energy. If the activation energy is too high, the reaction will not occur. However, when there is an enzyme, it can interact with the reactants and allow a different pathway with a lower activation energy, making the reaction easier to achieve. So the term substrate is what we call reactants that pair up with enzymes. The substrate will bind to an enzyme's active site where the reaction occurs and the products are formed. After the reaction, the catalyst by definition should remain unchanged and shouldn't be used up to repeat the reaction with new substrate molecules. In addition to the active site, many enzymes also have an allosteric site. This is a different site on the enzyme where if a substance enters an allosteric site, it has a chance to change the shape of the active site. And finally, it's important to note that the name of an enzyme often reflects its function. So you have a list there as shown. So for example, oxidoreductases are, from the name itself, involved in redox reactions. In the case of medicinal drugs interacting with enzymes, their role is usually an inhibitor. By being so, the drug may switch off an enzyme that is related to a certain disease or condition. So take for example this diagram over here. This is what we call an enzyme-substrate complex, wherein this shows your natural substrate binding with its enzyme. If the products of the enzyme can lead to something we don't want, let's say the products can lead to a disease or exacerbate a condition, a drug that can stop the enzyme can be helpful. Okay, now let's take a look at how a drug molecule can become an enzyme inhibitor. There are three ways. One is competitive inhibition. In this case, an inhibitor competes directly with the substrate for the enzyme's active site. If the inhibitor binds first, the substrate can't get in and the reaction is blocked. The second is non-competitive inhibition. This is when the inhibitor binds to not the active site but an allosteric site, causing a change in the enzyme structure that still prevents the reaction, even if the substrate is present and ready to bind to it. Last is uncompetitive inhibition. This inhibitor joins only when there is an enzyme-substrate complex. The binding of the uncompetitive inhibitor changes the enzyme's shape, preventing the reaction from continuing. Here is a list of drugs that target enzymes. You may want to pause this video to take a look at it more closely. Next, the last drug target that we are going to discuss is a bit different because these are not proteins, rather a different type of biomolecule, which is, as mentioned, your nucleic acids. Recall that your nucleic acids can either be DNA or RNA, and both of them are involved in the central dogma, specifically in the processes of DNA replication, DNA transcription into RNA, and RNA translation into protein. Drugs that target nucleic acids are usually antiviral and anti-cancer because viruses and cancer cells multiply using DNA or RNA. Now, there are 5 types of nucleic acid targeting drugs, which I will go over very briefly. 1. Intercalating agents are drug molecules which have the right shape to insert between adjacent base pairs and distort the DNA backbone, eventually damaging it. Second, topoisomerase inhibitors prevent unwinding of the coiled DNA so it cannot undergo replication or transcription. Third, alkylating agents add alkyl groups which interfere with the original IMFA within the DNA. Fourth, chain cutters make use of free radicals to damage DNA. And 5, Chain terminators prevent the formation of phosphodiester links, thus cutting the nucleic acids short. Now we've covered the 3 drug targets, we are now ready to explore how drugs are designed. Specifically, we will talk a bit about structure-activity relationships, pharmacophores, and Lipinski's rule of 5. Structure-activity relationship, or SAR, helps us understand the connection between the structure of a molecule and its biological activity. By studying SAR, we can identify which parts of a molecule are important for its function. SAR usually focuses on functional groups of a molecule as these are most likely responsible for binding interactions with a receptor. There are a few ways to study SAR. First, you can use X-ray crystallography if you can isolate the drug target and see the actual binding of the drug molecule to its binding site. Next, you can use molecular modeling as a more theoretical and computational approach. It may not be very accurate, but it is cost-effective and useful in predicting SAR nonetheless. And lastly, you can synthesize and test analogs. An analog is a modified version of the original drug molecule. It just has a different functional group. More on this on the next slide. Changing the functional group when testing for biological activity helps us identify which structural changes improve or hinder biological activity. If an analog will show a lower biological activity after modification, the altered functional group is essential. If the activity remains similar, then the altered functional group is not essential. And from that data, we can try to narrow down the cause of the biological activity to a few molecular similarities and establish a pharmacophore. So suppose we study the hypothetical drug molecule glipin, shown here. We can create 3 analogs, for example, with slightly different functional groups, such as these ones. Okay? And check which functional group is related to the activity of the drug. Now, this may need lots of lab work. However, eventually, you will be able to identify a pharmacophore. Once the pharmacophore is identified, researchers can now focus on the important functional groups and modify them to reach the best version. A 2D pharmacophore will show you the important functional groups, while the 3D pharmacophore will show you the bond angles, bond distances, and the overall orientation of relevant functional groups in space. But for limitations of pharmacophores, I'd like to emphasize these ones. Pharmacophores emphasize functional groups and might downplay the role of the carbon skeleton that contribute to possibly crucial van der Waals forces and hydrophobic interactions. Also, the size of a molecule and its ability to fit within the binding site is another important factor that the pharmacophore does not address. A functional group that is part of the pharmacophore might not be essential if the drug can form alternative binding interactions with the target. Okay, next slide. Here, I would like to add more considerations in designing a drug, specifically a drug that you will take orally. So this is Lipinski's rule of 5. It is a set of guidelines used to predict the likeness of a compound to a drug. So even just by looking at a compound structure, according to Lipinski, we should be more or less be able to judge if it will be a good oral drug candidate or not. If the compounds will not satisfy the rules, it will not be a priority in research studies. Okay, now, what does this Lipinski's rule state? According to Lipinski, a potential oral drug should ideally satisfy the following. The molecular weight of a compound should be below 500 daltons because too large a molecule may often face challenges in absorption and distribution in the body. So we want to limit their size. So if you look at the illustration below, so you have here a magnified lining of the intestine. So your oral drugs will have to be absorbed and have to go through many barriers in the intestine. So there are considerations such as the molecular weight. So we want a molecule that is not too large for it to be absorbed in the layers here in our intestine. Okay. Next, log P. Log P is a coefficient that indicates fat solubility of a compound. The higher the value, the more hydrophobic a compound is. We don't want a value higher than 5 because that means it is not water soluble enough to be dissolved in the aqueous environment of our cells, and thus our body again cannot absorb it well. Third, hydrogen bond donors. This is simply the number of hydrogen atoms that are involved in hydrogen bonding. Lipinski's rule suggests that having 5 or fewer hydrogen bond donors is good, as too many can increase polarity and reduce the compound's ability to pass through the lipid bilayer of our cell membranes. Fourth, hydrogen bond acceptors. This is simply the number of oxygen and nitrogen atoms present. The rule suggests fewer than 10 hydrogen bond acceptors is good, the same reason as the previous one, wherein we don't like compounds that are too polar. Again, because the lipid bilayer might not let it pass through. Lastly, rotatable bonds. These are the single bonds that are around sp3 hybridized carbons. Too many bonds that can freely rotate may decrease the compound's capability to form stable interactions in the body. There is a certain rigidity expected in the structure of the molecules in order for them to activate the binding sites of receptors via induced fit. So the rule recommends fewer than 10 rotatable bonds to maintain appropriate molecular rigidity and stability for drug action. So if you're wondering why it is rule of 5, 5 there just means that all the numbers that are in the rules are divisible by 5. Lipinski's rule of 5 is a useful predictive tool for the drug discovery process. It is cost-effective to limit drug candidates early on based on these rules. However, this only predicts whether an orally administered drug can be absorbed by the body, and it does not ensure the efficacy and the side effects of it. So further testing is of course required. But what we are sure of is that this set of guidelines give us insight in the desired structure and binding interactions of many drugs. So far, we only talked about how scientists find potential drug candidates, but this is only at the starting point of drug development. From around tens and thousands of drug candidates, less than 1% will be approved by the FDA and be manufactured to be available to the public. So at this point, I will be showing you a video clip just to have a brief overview on how drugs from the laboratory get to the public. So to run through drug development, it starts with a chemical synthesis or structural modification of drug candidates that show promise in having therapeutic activity. Next is preclinical testing, where effects on body functions, mechanism of action, and toxicity are tested. Next is the clinical trials, where the drug is tested on real people. So efficacy is investigated and adverse effects are monitored. Not all drugs will pass preclinical and clinical tests, but for the very few that do, they can be approved by the FDA for public use. And continuous surveillance and monitoring of such approved drugs will be conducted under Phase 4. So we've covered everything in this outline. So that concludes our video, Medicinal Chemistry Part 1. Thank you all for listening.